Good morning from College Station. William Neal with you, with the local folks on the 13th of November. Looking at our class archive site, and I was explaining to the folks here in the room that the technology is sort of teetering. You know, I'm having a lot of difficulty getting these files uploaded to this class archive. Something is causing the WFSC dist-ed server to choke down. But I think we're up to date there. Here we are on the 13th, and the plan is to consider today issues having to do with activity patterns of fishes, and then move on and talk about some biotic components of habitat that I haven't dealt much with to this point. I explained in an email that I sent out yesterday afternoon that I intend to forego a live session in lab this afternoon for Lab 6. I've been focused on other things. You've probably been focused on other things. We're all tired. And most of you are assuring me that you would really rather have the option to speed things up a little bit rather than doing the live thing. So I decided after I played back the archived files from 2011 that I wouldn't be able to do any better, and that it would be more efficient for all of us to do it this way. So there they are. They're already posted. I've converted the AVIs after some slight revision to MOV files, so they're on the server. There's two files that are sequential, A and then B. The handout is identical to the one from last year, but it does have a date to reflect the fact that it's current for fall 2012. And the presentation, the black and white version, well, it's actually black and white and it's outlined, but there's some color photos in it that make the file, unfortunately, a little large. That's there as well, but it's still got a 2011 date on it. And here's the simulation model that I use in this presentation. As you all know by now, today marks the end of having to produce something to turn in lab for undergraduates. Some of you may be a little behind, but I'm sure you're working hard to get there. Graduate students need to be working with me and maybe with each other in some cases to pursue a project which is going to be the focal point for lab for the remainder of the course for graduate students. I do require and hold responsible all of you, undergraduate, graduate, live and local or distant, for actually going through Lab 6. It is sort of the integration session in which I talk about the foundations of ecofish, how it came to be, present some of the experimental studies that are the basis for the structure of ecofish, and talk about some philosophical issues that I think are important having to do with modeling. So some of that may be subject to coverage on the final exam, but that's the place where it will be important for you to have command of some of the material. I won't cover little nitpicking things, but the broad brush ideas that come up here could be subject to coverage on the exam. I might ask you simply, as one of the 5 final exam questions, I'm not sure I'll do this, but I might ask you, what do you think about ecofish? Do you think it's a valid approach to understanding? It's not a research tool, but it is a teaching tool, and it's based on research. So, I don't know how I could phrase that so that it would be a doable deal for you. I also talk about a simulation of a variant of the Lab 6 model, which is WFSC417617. It was revised in 2008. And talk about it in the context of something I'm going to bring up today, which is the energy bonus idea. So do that either here in this room if you're local and want to work on the computers here, or do it at home, or do it wherever you are, but do carry out the lab. I'll be available this afternoon in my office to meet with anybody that would like to talk about anything, but I'm thinking particularly about graduate students and their projects. Both local students, you're welcome to drop by. Distant students, you're welcome to send me an email and we can get together and agree to talk on the phone or do Skype or whatever's appropriate. So that's the plan. You know, really, the plan is pretty much to wrap things up, you know. I know you're going to miss these little computer side chats, but they're going to come to an end, and my goal is actually to have the last session Thursday. I've got 2 segments of material to talk about today, and there are 2 more that will finish the course. And I think I can do that today and on Thursday. It might be necessary to come back on Tuesday of next week, but I'd prefer not to do that because that's Thanksgiving week and everybody's thinking about trying to get going someplace. So anyhow, we're gonna shoot for doing that. Let me leave the archive and go instead to this PowerPoint presentation. And I've handed out the usual 3-slide per page black and white version of this to local folks, as well as the next presentation, which I also intend to cover today, as well as the doc file for Lab 6 and the black and white presentation file. So I want to consider with you activity patterns, and by that I mean movements around and movements in the habitat, within habitat movements. And first I'll just talk about the levels of locomotory activity that are observed in fishes. And this is based on, you know, sort of lots of field observations by lots of folks. Some of it pretty anecdotal, some of it fairly experimentally well designed. But it looks like the diel patterns that occur in fishes and their activity levels tend to be obviously correlated with light and temperature regimes and their cycles, maybe to a lesser extent with dissolved oxygen cycles. The peak activity levels tend to occur at times when the animals are actively engaged in foraging. You know, that's why they're moving around, is to feed, typically. And most folks will distinguish groups of fishes as being either fundamentally nighttime or daytime in their activities. Nocturnal fishes are thought to rely primarily on olfaction in their, or chemoreception at least, if not olfaction, in food finding. And lots of, you know, here are some examples: sharks and gars and bowfin and the anguilliform eels. Catfishes, carp, use their senses of chemoreception to find food and to distinguish between food and non-food. Diurnally active fishes are more visually oriented, and fishes that we think of as being truly schooling, and in the case of freshwater fishes, even ones that aren't truly schooling like the herrings, but things like the shiners, the Tropus genus, for example, and the minnows. Salmonids, under many conditions at least, Athereniforms, the silversides, sunfishes to some degree, perches, cichlids, and reef fishes all are sort of diurnally active. But within these groups, there's a lot of variation, and a lot of that variation is size dependent, and I'll get to that. A lot of them are actually not so much nocturnal or diurnal. They're actually crepuscular, meaning they're active at the interfaces between day and night, either at dawn or at dusk. And these fishes are said to be crepuscular. Activity levels tend to correspond in many cases with movements within the habitat, from places of rest to places of foraging, or between refugia and foraging sites. And you can distinguish several different categories of this sort of thing. Offshore, inshore, for example, is a common pattern observed both in marine and freshwater habitats. Some fishes tend to move offshore during the day, and then at night they come inshore. And a lot of the sharks have this pattern of behavior, which is one good reason maybe you shouldn't go swimming in bays at night or in the passes, because that's when the sharks are coming into shallow water. A lot of the larger freshwater species, the adults, the large-bodied forms, tend to have this pattern of being offshore during the day, or at the edge of the limnetic zone in lakes, between that and the lacustrine, which is the open water. I'm thinking that through in my head, and I don't like the way that sounded. How about littoral zone, the edge of the water, the weeded part of the lake, and the open or limnetic part? I think that's all of that is lacustrine. I mean, that has to do with lakes, but they've got the littoral zone near shore, the shallow water that's often vegetated, and then the limnetic or open water region outside. And fishes like, for example, adult bluegill in north latitude lakes tend to show that pattern of movement. You just don't find the big fish too often inshore during the daytime. They're mostly at the edge of the weeds or beyond, and then come in at night. On the other hand, there are other fishes that have just the opposite pattern of movement, tending to come inshore during the daylight hours and live in the weeds, and then move offshore at night. And a lot of the same fishes that have the other pattern during as adults will reverse that pattern when they're young. For example, young bluegill tend to come in during the day into the weeded zone and then move offshore at night. Just the opposite. They sort of pass in at dusk, I guess. And to a certain extent, carp do the same thing. Juvenile carp will be found inshore during the daytime, and no big carp, and then at night just the opposite happens. You get the big carp in shallow water and the little carp are gone. A lot of the invertebrates that I talked about earlier, I gave you the example of yellow perch feeding on hyalella. Invertebrates like hyalella tend to adopt that pattern of being inshore in the weeds during the day and then moving out into the open water at night. And most folks believe that this kind of pattern and its reversal between as the fish pass a certain size probably relates to feeding and to avoidance of being feed, predation. Sort of a complementary type thing is up and down in the water column and out in the open lake or ocean in the limnetic zone. A lot of open water fishes and invertebrates come up at night and feed, or up at dusk and at dawn, and then down during the day. So down during the day, up at night is a common pattern in limnetic zones of lakes and in open water of the sea. Some folks have thought about this a lot and tried to account for the existence of this cyclic movement up and down and the timing of the movement, and have concluded that it may be partly a matter of trying to avoid predators, or maybe trying to avoid light damage to sensitive eyes. If you came up at high noon, it might be so bright that it would be hard on the animals. Maybe pursuit of animals that are doing those things in the form of following vertically migrating prey. And then finally, there's the idea that this has to do with maximizing energy retention, or the energy bonus idea. I attribute this to work by a couple of Canadians. Ian McLaren, I think, was the first person to suggest this, at least to my knowledge, in print. He was at the University of Toronto and worked on he was in Ontario, the Fisheries Research Board of Canada. I think he was also associated with the University of Toronto. And McLaren developed the idea to account for the vertical migration of zooplankton in northern lakes, the diel migrations. And what he argued was that these things move up toward the surface and feed in the surface layers of the lake, and then go down during the daytime and do their food processing at depth where it's cooler. And that the lower temperature at depth means that more of the energy that was in the food that the zooplankton consumed gets retained as new zooplankton instead of being wasted in respiration. J.R. Brett on the other coast of Canada, out in British Columbia, took McLaren's idea and applied it to sockeye salmon movements up and down in the water column. And it made a pretty convincing argument that, whatever the cause for the timing of the cycle, it's advantageous to feed where food is abundant and where you can see the food if you're a visually oriented fish. But then go down into the water column and sort of do your resting and digesting and food processing at depth. I kind of have a little graph here to illustrate the points that I just made. Here's depth going down and temperature coming up as we go toward the surface. And over here is gross growth efficiency. And so the idea is that at midnight and during the night, the animal is at depth, but it rises at dawn and dusk and does its feeding when it's efficient to feed, when it can see the food, but the food is abundant and there's not too many things around to eat it. And then it goes down in the water column, and during the day is deep, where the gross efficiency of food conversion is higher. That's the argument that J.R. Brett made, and he followed on the heels of Ian McLaren. Other people have wrestled with that idea, and I wrestled with it a little bit with ecofish to see what ecofish would do with a cyclic regime. And I present the results of that wrestling match in the presentation in Lab 6, so I'll leave you to find out for yourself what I think might be going on. It's a little different than the arguments that these guys made, but I can cut to the chase and tell you that ecofish does achieve an energy bonus by having a diel cycle of temperature. So that's the diel situation. What about a seasonal situation over the course of the year? And there, if you're talking mature adults, then one of the periods of intense activity is during the time of spawning and just ahead of spawning. If you're talking fish in general, juveniles, more activity occurs during the warmer periods of the year associated with the Arrhenius effect. And in lower latitudes with wetter times of the year, when the floodplains of the Amazon are full of water, that's when the fish are really aggressively out, moving around, feeding, getting ready to spawn or spawning. So activity is greatest during warmer and wetter seasons. In mid-latitudes, though, in the plains of Oklahoma and Kansas, where it really gets warm in the midsummer time or in August and July, there may be a depression of activity, sort of the dog days effect. Activity may drop off and less feeding and less growth. And I once did simulations with ecofish to demonstrate that too, but it wasn't really all that convincing, so I dropped that from being a showpiece. I'm not sure I did it right. So seasonal movements that amount to migrations also occur. In the case of freshwater fishes, the pattern that is regularly seen is for the fish to move out of shallow water during the wintertime. I'm talking now about mid-latitude and more northerly in the case of northern hemisphere fishes. The sort of thing you would see in lakes or rivers in the northern part of the U.S. Lake and reservoir fishes tend to basically abandon the shallow water part of the lake during the coldest part of winter, move into deep water, move offshore. And stream fishes, salmon included, tend to go downstream and leave the headwaters regions where the water is shallower and move down into the deeper parts of the downstream reaches of the streams. Marine fish tend to move poleward during the summer and then back toward the equator in winter, giving it an annual cycle of distribution, and probably some of them, maybe all of them, get help from the ocean currents. And I think I already alluded to the movements of skipjack tuna in the Central Pacific. You know, they sort of follow this loop, and the loop is sort of directed and assisted by the currents, the prevailing currents, the Kurashil, takes them across the North Pacific. And then they come down through Hawaii during the late summer and fall, and then in the wintertime they're down around Christmas Island, and then back north through the Japanese home islands and back north again. The North Sea herring, their movements may have a circular motion too, but what you see is simply a pulse, where in summertime the distribution spreads out and the edge of the distribution moves north into the North Sea. Way up north, I mean, we're talking 75 degrees north and further. And then in the wintertime, that distribution sort of moves south and clumps again in the region between Iceland and Norway, so that the winter distribution is just the southern end of the whole summer distribution. Wrote a paper one time talking about the fact that maybe movements like those demonstrated by skipjack and certain salmonids may be originally entrained as environmental regulatory movements, reactive type movements that then become predicted, and after a while the movement is entrained, and it would occur even in the absence of any seasonal change in thermal regime. But probably, in the case of albacore and fishes like that, you can clearly see a shifting in the northern limit of the distribution between warm years and cold years, and certain isotherms tend to be boundaries for that distribution. What I have been spending some time on lately, and I'm still not completely happy with the result, but I think I'm getting there, is extending the little models that I showed you at the end of the last session, where I talked about reactive movements, reactive regulation in gradients, to a couple of other ideas. One being taxis, asking the question, could I take the little model that I showed you, the variants I called orthokinesis, clinokinesis, and clinokinetic avoidance, and convert that to a model that would allow the virtual subject to react in a taxic way to an environmental stimulus. And that was pretty easy. It was a little harder when I asked the question, could I make the virtual subject deal with the world in a predictive way? In other words, would it be possible to have the virtual subject learn the value of habitat in different places that are available to it, remember those values in a way so that it can move in a predicted way to the place where habitat is optimum, rather than moving with optimum habitat? See, those are two different ideas. And I have something that's working pretty well that I hope to finish up, and if I do, I'll post a little presentation and show you those models before we're done. Okay, so I think that's the end of that show. I want to move on now and talk about the next segment of the material. And this has to do with a part of the habitat that I really haven't talked about, a part of the environment, and that is the biotic part. You know, I guess we talked about feeds and feeding at one point, you know, and forage, but we didn't really talk about the other biotic components, the opposite in the predator end, for example, or pathogens and parasites, fish disease. And so I'm going to, you know, do a really cursory trip through this subject, partly because we deal with it in other places a lot more effectively than I'm going to deal with it. I think several of you are probably taking Dr. Gatlin's course on fish diseases right now. Partly because I just don't know a lot about the topic, and I don't know how to deal with it quantitatively, which is, as you know by now, the way I like to deal with things. So I want to at least consider, though, these problems that fish face of pathogens, parasites, and predators. And the first thing to make clear, I guess, is the way I see it, the boundaries are not distinct between or among those groups, between pathogenic disease and parasitism and predation. Sort of thinking about it, and I'm not sure that I have a lot of company on this way of looking at it, but I think it's reasonable to think that pathogens and parasites can be distinguished from predators in that they sort of attack at a fairly slow pace and may not actually kill their host. So, you know, pathogens and parasites attack without necessarily killing, and the damage tends to be done over periods of hours or days, depending on the system. Pathogens do their damage by typically some toxic substance that either the pathogen itself produces or something that it stimulates the host to produce. Parasites, on the other hand, just sort of eat the host a little bit at a time, eat it gradually. They consume the actual tissues of the host. And then I think of predators as sort of you know, an extreme there in terms of attacking and consuming host tissues, and that they do their damage in short order, typically a matter of seconds or minutes maybe. Sometimes they will kill the prey item and then consume it. Other times they'll consume it and the prey item is killed in the process of being, or after consumption. In other words, a predator may swallow its prey alive in many cases. So another way of sort of thinking about the boundaries between disease and parasitism and predation is to apply Fry's physiological classification of environment. And as soon as you do that, it becomes pretty clear pretty quick that Fry would argue, I don't think he actually did, but if you, I'm sure, brought him back and asked him, I think he would agree that pathogens and parasites tend to load the system or act as masking factors. That was one of Fry's categories, and you'll find out about the categories when you play back the Lab 6 presentation, or if you've already looked at the paper that I did. So Fry would say that pathogens and parasites are masking factors in that they obligate the animal to do extra metabolic work just to maintain itself over what it would have to do if the loading factor were not present. So it's easy to think about. You know, you're going to have to do more work to support yourself and a parasite than just yourself, presumably. The metabolic capacity that you invest in something that's more useful to you, like growth, you have to spend supporting the parasite. So parasites and pathogens are masking factors. Predators, on the other hand, are clearly lethal factors. I mean, they're just as, in fact, I think predators are better examples of lethal factors than some of the physical chemical factors that are traditionally offered up. For example, I have a hard time really thinking about low oxygen as operating as a strictly lethal factor. It's a limiting factor, and it can go to lethal extremes, but it doesn't abruptly destroy the machinery that is metabolism the way a predator does, or a toxic substance like some neurotoxin that's secreted by some organism. So I would call predators lethal factors and pathogens and parasites masking factors. Something we're not going to get into at all because, as I said to you from the beginning, this is an audicology course, not a synecology course. But if I were talking synecology, I would argue that at the next level above the individual, when you start talking cohort or population, lethal factors become loading factors. Think about that just a minute with me. I mean, you can see the logic there. We wrote a paper a number of years ago. Myself, I was first author. Kirk Weinmiller in this department was a co-author, and some other folks in the Netherlands, and John Miller in North Carolina. And what we argued was that these factor classes get translated as you move up the hierarchy or down the hierarchy. Maybe I'll talk about this in the last session, but our argument was that lethal factors for individuals, factors that completely destroy the integrity of the system that is the individual, don't destroy populations. They kill the individuals in the population, thereby obligating the population to work harder to support itself, to produce more individuals to replace the ones that have been killed. So in that sense, lethal factors operate like loading factors at the level of the population. I thought that was a neat conceptual leap. So what kind of things are pathogens? Everything you can think of, Everything likes chicken. Well, everything likes fish, too. Viruses like fish, and I've got some examples here, and all I got up is names. You have to tell me what they are. You probably learn all about them in fish diseases, but infectious salmon anemia, ISA, big problem. Channel catfish virus disease, red spot in carp, lymphocystis, a little thing that causes these lesions on the skin, and not really dangerous, but you see it a lot in aquarium situations. Bacteria, and these are the scary ones, bacterial hemorrhagic septicemia and furunculosis in trout, columnaris. And then you got things like saprolegnia, which is a fungus and sort of cottony stuff on the skin of the fish. And a lot of people say, well, that's secondary, the fish has already got a problem caused by something else, and the saprolegnia amounts to a secondary problem. Algae. Some algae can actually be pathogenic, but most algal problems occur because of toxins that are secreted by free-living algae. Things like red tide, the dinoflagellates are notorious for producing toxins that, when in the water, have adverse effects on fish and other things. In fact, on you, if you've ever been down on the coast during a big red tide bloom, you can smell it and you can feel it. It burns your eyes. Blue-greens, golden algae that are in freshwater mostly. The dinoflagellates like brown tide, or red tide, I guess, more than brown tide. All those secrete toxins that have adverse effects. Protozoans. Last things I looked at, sort of reviews, suggested that protozoans are the group of things that are most often responsible for problems in fish culture. And some of the bad actors are things like the ciliate ick, or ichthyophthirius, white spot disease. You see that often in cultured fish, often in aquaria. Amylidinium, everybody knows and fears in the culture of things like red drum and hybrid bass. It's a gill parasite. particularly a gill parasite. Costia is another flagellate that causes big problems in fish culture, particularly salmonids. And then you got sporozoans that I don't know a lot about. That's another group of protozoa, Mixobolus and Lentospora. And from what I read, those are the biggest problems in wild fish. So wild fish, sporozoans are the greatest protozoan threat. But in cultured fish, it looks like, it's mostly ciliates and flagellates. Metazoans, things that have more than one cell, everything else. And I start with trematodes. I maybe should start further back, but I don't know where. Trematodes are the flatworms. They come in two different major groups, and Dr. Dronin could tell you all about this. He's a specialist. There's the monogenetic trematodes that have a direct life cycle, one host. And they tend to be ectoparasitic. They tend to be parasitic on the gills of fish. And then there's the more complicated forms. The digenetic trematodes are endoparasitic in fish typically, and they have multiple hosts. And something, eats the fish, and a bird eats the fish and gets one of the life stages, and then usually it matures, and then the fecal material is the parasitic egg, I guess, or hardened life stage that's eaten by the fish, developing, I don't know what these things are. They're not just, I guess they're eggs, maybe larvae. I forget the names. At one point I was kind of into this with a student, and we had it all worked out. And, we were talking about a thing I'll get to here momentarily. Anyhow, digenetic endoparasitic trematodes and monogenetic ectoparasitic trematodes. And then there's some other worms, things that are generally thought of as worms, but not segmented animals. Things like cestos they're segmented, but they're not annelids. Anyhow, cesto, tapeworms. Everybody knows what a tapeworm is. And nematodes are roundworms. And then spiny-headed worms are the acanthocephalans. And I know of at least one instance where spiny-headed worms nearly wiped out a crop of redfish down on the coast here in Texas one year. They're not normally that conspicuous in there being so much a problem. It's usually amylidinium. Leeches. Leeches are annelids, I guess. Are they not? Somebody know their taxonomy? I think they're annelid worms, segmented. And leeches not only will, act like leeches and attach to a host and suck tissues out, but they also act as vectors for carrying other parasites, like Lymphocystis, for example, is transferred from one host to another by leeches. Mollusks can be parasitic. The larval stages, the Glochidia. In fact, they can wipe out fish under certain conditions. There's this tremendous outpouring of this glochidial stage, and they come up and they clamp onto individual gill filaments, and they can literally cover the gills of fish. Copepods can be parasitic, and it's not uncommon to find fish, and it seems to be more problem in sort of polluted waters. I don't know why that is, but leeches and copepods, they're conspicuous. You catch a fish and you can see copepods hanging off its eyebrows. Seems to be I call it eyebrows, the orbit around the eye will have a couple of copepods attached. Isopods are other crustaceans, and I think all the truly parasitic isopods, I believe, are marine. I don't think there are any freshwater parasitic isopods. And then finally you reach the level of chordates, the lampreys and the hagfish we've already talked about. And there's some teleosts also that are parasitic. Defenses against pathogens and parasites begin with the antibodies that are familiar to us in dealing with human disease. And they come in several different categories depending on how they act. Antitoxins and precipitants that cause precipitation, and lysins that split, and agglutinins that clump, I guess. I mean, these names are more labels, I think, than really that helpful. I don't know much about it. Mucus is kind of a special defense that fishes have that we don't think much about as mammals. We got mucus, but it's usually not covering our entire skin surfaces. But in the case of fish, that's sort of the first barrier against attack by all kinds of things. It's also important, we've already considered in terms of osmotic issues. But one of the things that happens when you handle fish is you break that mucus layer, and that exposes the fish to attack by things like bacteria and fungi. White blood cells, scavenge blood-borne foreign bacteria and things like that. And then there's some, more interesting to me at least, things that involve behavior, things like cleaning symbioses. And what these amount to, and they're very highly developed in fishes and other aquatic organisms, is cooperative arrangements that go on interspecifically. Sometimes, interphylum almost between different organisms. And you've all seen the Discovery Channel and Nature Channel footage on cleaning symbioses, so it's hard to impress anybody by talking about that. What it amounts to is fish like groupers, for example, that usually get featured in these coverages, will come to a cleaning station that's occupied by a wrasse or a butterfly fish or something, usually a wrasse in the case of groupers. And everybody understands what the game is. The grouper comes up in a very humble way and presents itself and doesn't threaten the little fish at all. The little fish come boldly out and swim into the grouper will sort of lie on its side and open an opercle and allow its gills to be exposed, and the cleaner fish will come in and sort of nip and clean. And it's really a pretty impressive thing when you think about what had to be involved for that kind of complex interaction to develop, from scratch. I mean, probably at the beginning, the little wrasse was going to get eaten by the grouper. And that's what he was thinking about. Over 30 different fishes, probably a lot more than that. Mostly wrasses and butterfly fishes have been identified as having specialized cleaning symbioses. And then there's some shrimps that tend to be specialized cleaners as well. As far as I know, all of the real well-developed cleaning symbioses in fishes are freshwater. Are marine, not freshwater. They're almost all marine. They often are coral reef type situations, tropical. At this point, I'm always reminded, I was talking about this one year, and the very day I talked about this to the class, I happened to see a thing on one of those shows on Nature TV, Discovery Channel or Nature Channel, and it involved a totally different, but still clearly cleaning symbiosis that involved mammals. And some of you may have seen this. If you haven't, I hope you'll. It may be on YouTube someplace. You might be able to track it down. It involved a bunch of warthogs that came into this jungle clearing where there was a mongoose tribe. And I thought, whoa, these mongooses are going to be in trouble, But no, those warthogs came into the clearing and were very gentle, very passive, laid down. Mongooses started working them over, eating ticks and whatever, So it was clearly the same kind of thing that goes on in fishes. And I didn't realize that those kind of things happen in mammals, but interspecific cooperative relationship. very well organized. And another topic that's kind of near and dear to me that I've done a little work on, never as successfully as I would like, I call it therapy by behavioral regulation of environment. Therapy. And logically, you know, it's obvious to anybody and everybody that resistance to pathogen attacks ought to be maximal where the fish lives under otherwise optimal environmental conditions that maximize metabolic scope, because you got more to be loaded before you run out of capacity there. So, you know, it ought to be finding otherwise optimum environmental conditions should be really at a premium if you're under attack by something. But the thing that I don't think occurred to folks initially is that if fish are sick, then maybe they ought to go to subsets of environment that would otherwise not be optimal, if in fact that confers some advantage to the host over the cause of the illness. And the modern theory of, you know, I think still is that fever, physiological fever, amounts to a host defense mechanism whereby you raise the set point body temperature, which may be not so good for you, but it's even worse for your pathogen. And it looks like fish do the same thing behaviorally. Some very clever experiments demonstrated that if you inject, I think it was bluegill or some other sunfish species with Aeromonas, that caused the fish to increase its temperature preferendum by a couple of degrees. So it came to be known as behavioral fever. You know, I think, unfortunately, nobody's ever completed the circle on that idea. In other words, it's been shown that you can stimulate fish to change environmental preferences by introducing a potential threat like a pathogen, a bacterium. But I don't think anybody has shown that it actually is curative, that the increase in the temperature selected, for example, feeds back to improve the host's chance of beating the disease or beating the pathogen. We tried to do it, a couple of former students and I, and the system we focused on was ick and mosquito fish. And the reason we chose that system was because mosquito fish have a temperature preferendum that's just right. It's about 27 degrees for mature females. And ick is treated commonly in aquarium situations by raising the temperature to about 80 and 82 or something. And I'm not sure, but anyhow, it worked out that was about 2 degrees greater than the basal preferendum of the mosquito fish. And so our hope was, hey, you know, we're going to infect mosquito fish with ick and then demonstrate that infected, or test whether or not infected mosquito fish will select higher temperatures in a temperature preference situation than healthy ones. And then what's more, we're going to show that in fact that behavioral therapy works, that the mosquito fish actually are better able to overcome ick, because we thought that was a given, you know, because it's already been shown that if you raise the temperature, ick tends to be controlled. Didn't work. Why didn't it work? The sick mosquito fish wouldn't do anything. We could never get them just sick enough. They were either perfectly healthy or they died. And they wouldn't respond. They wouldn't behave, you know. We tried the same thing with amylidinium and red drum. One of our recent, Dr. Dronin and I had a co-chaired student that tried to do a similar experiment with amylidinium and red drum, and there we were looking at salinity, because salinity is a environmental treatment for amylidinium. Didn't work for the same reason. They either got sick, Valdemar, and died, or they didn't get sick and didn't care, you know. So those experiments still are out there to be done by somebody that's more capable or more lucky than we were. But I think it's a neat idea that a poikilotherm might engage in behavioral fever. A final idea along these lines, and I think this is pretty well documented in the literature, is that Euryhaline fishes, mullets particularly, may engage in a form of behavioral therapy that involves a voluntary salt treatment. When they get loaded with freshwater parasites in the fresher part of the habitat, then the thing they do is go into the saltier water, and that wipes out their freshwater ectoparasites. And then they get loaded up with marine stuff, and they move into freshwater and unload that. And people who have collected things like mullets regularly find that individuals are pretty, discreetly parasitized either by freshwater forms or by saltwater forms, sometimes individuals in the same school. This individual got a load of saltwater parasites, this individual got a load of freshwater parasites. And I think that's given rise to this idea that maybe they're going back and forth and sort of washing themselves. But, again, I don't know that anybody's experimentally demonstrated that mullet or any other fish will voluntarily change its salinity behavior in response to ectoparasite load. So, predators, everybody knows about predation. What can I tell you? Everything eats fish. And the predators that are important to fish range all the way from the most primitive forms. I guess salinirates are my, most primitive. Like hydra, for example, is a terrible predator on little larval fish in fish culture sometimes. And then at the other end of the spectrum, taxonomically, there are birds and mammals that are obviously very predatory on all kinds of fish. And, what can you say other than that, everything happens. Defenses against predators. I would argue that the best defense against predation is to be someplace where the predator isn't. At least when the predator's not there, even if it's there sometime. And I think a lot of this switching inshore and offshore and up and down in the water column has to do with predator avoidance. If you can't do that, then try to make sure that the predator looks at you and doesn't see food. Not like the coyote and Gordon Gecko in the recent ads. Have you seen that ad where the coyote comes running up and Gordon Gecko's there, and in a coyote's mind, what he's seeing is a little roasted gecko on a platter, So you want to avoid being seen as food by the predator that's a threat. And some ways of doing that are having protective form. And probably, the sargassum fish would be the best example you can imagine of having within fish world. I mean, you can think of stick insects and things that don't look like insects. But within the fish world are sargassum fish. I've picked up a clump of sargassum, and there was the sargassum fish until I shook it and it flopped out on the ground. I never saw it, Protective form, coloration usually goes with the form, blending in with the background. Protective behavior, being cryptic one way or the other. Mimicking something that's noxious, and I don't know any really good examples. There's probably some good examples among the wrasses of looking dangerous, or something that looks like a moray but isn't, like something looks like a monarch butterfly but isn't. It's a viceroy, And then schooling is considered by a lot of folks to be essentially protective behavior. And in fact, it's pretty easy to demonstrate that if you frighten a loosely gathered school of fish of certain species, they come together in a clump and form a very dense school. And the human interpretation of that is that a dense school of individual fish resembles some superorganism, a bigger fish that could be something you wouldn't want to tackle if you're a predator. A lot of fish have spines and armoring of various types. And if you don't believe that, try taking a bunch of little yellow bass out of a gill net on a freezing night. You know, sometime without gloves, they'll eat your hands up. So spines on the scales, spines on the fins, spiny rayed fishes are thought to be a protection against being swallowed by something. Poison glands, some fish have poison glands. First, propenoforms are really notorious for that, and then some fish generate powerful electric shocks that can be defensive, I guess. And I hate to just dead end like that. That's the end of what I got. That's all I got. And so we will stop here today. I hope you will complete your work on Lab 5, and then at your leisure, in the not too distant future, at your convenience, probably not at your leisure, review Lab 6. You won't have to do anything except kick back and enjoy and think, I hope. And then I'll be talking with you individually, I hope, about your projects. And then we'll wrap up, I think, on Thursday as far as the lecture session goes. So we'll stop for now.